So I'm going to talk to you today about reconstructing the evolutionary history of cancer in individual patients. And unfortunately, at the outset, I have to offer a disclaimer. There are no neural networks in my talk, but I'll give you some fun Bayesian stuff anyway. So I want to tell you about the computational work we've been doing, but I first have to tell you a little bit about genetics. So your body has 37 trillion cells in it, and you can think of every cell as this cute little nanomachine. Now, the genome is the program that dictates how that cell works. So if I'm talking about the genome, you can represent it as 3.2 billion letters drawn from the set ACGT. And if you have 2 bits per letter, then your total information capacity is about 1.5 gigabytes. Now, every cell in your body carries an identical copy of that genome. And at left, what you're seeing is this flourish ceiling bookcase at the Wellcome Collection in London. And so they actually have a printout of the whole human reference genome there. So it's just filled with little tiny A's, C's, G's, and T's. So this is about 5-point text. If you opened any of those books, that's what you'd see. So, of course, I just told you that every cell carries an identical copy of the genome, but that's a lie, because it's an almost identical copy. The reason I say almost is because you have mutations. So your cells acquire mutations when they divide. You get about 1 mutation every division. And if you run the math, we say every cell divides on average about 100 times throughout your life. So if I'm referring to a mutation, you can think of it with regard to the black bar here. It's saying if I were to crack open volume 19 of the book on that shelf, and I were to flip over to around the 27 millionth letter, maybe I changed the A to a G. So if we do the math, we say that we have on the order of 10 to the 13 cells, that's the 37 trillion, and you have 100 divisions occurring per cell through your lifetime, 1 mutation per division. That means you have about 10 to the 15 mutation events in your life. Now, to me, this is crazy, because as I told you, you only have on the order of 10 to the 9 positions in your genome. So what that implies is that somewhere in your body, every genomic position is getting mutated more than 1 million times in your life. And so I think that speaks more than anything to just how robust and how beautiful the body is as a biological system. So normally I talk to biologists, but I'm really happy today to have the opportunity to address a room full of computer scientists, because it means I get to sell you on my favorite idea, which is that cancer is an optimization problem. So we're just talking about mutations. Cancer is caused by mutations that allow your cells to replicate uncontrollably and invade surrounding tissue. That's basically the definition of cancer. We evolved from single-cell life, where you had all these single cells competing against one another. And so today, of course, you have mechanisms that allow your 37 trillion cells to cooperate together as a single cohesive organism. And that's good, but they've been grafted on after the fact. It's kind of a hack. And they can be defeated by mutations. Mutations can knock out those mechanisms. So, as it turns out, you only need about 5 mutations in any given cell to turn a normal cell into a cancer cell. Now, this is dependent on tissue type. For brain, we might say it's 6 mutations. For breast, we might say it's 4 mutations. But about 5 is the right number. And so there's also thousands of mutations that are basically noise because they don't affect how the cell operates, but we'll ignore those for now. So I like to think of cancer as a result of this massively parallel search through mutation space for the right combination of mutations necessary to render a cell cancerous. So let's start at that point. We have the 5 mutations that you need to turn this pink cell you see at the left from a normal cell into a cancer cell. But of course, the cancer is growing rapidly. It's dividing rapidly. That means that as it grows, all the cancer cells are acquiring new mutations. And so you need to think of cancer not as a single homogeneous mass, but actually as this whole set of different species of cancer cells that exist in the patient, all competing against one another. So the different colors of cells you see here correspond to genetically distinct subpopulations of cells, each of which has its own characteristic set of mutations. So imagine that the cell's been dividing. We have all these different types of cancer cells emerging in the patient. The patient is getting really sick. He goes into the clinic, is diagnosed. We begin treatment. So that's demarcated by this red line you see here. We start treating with a drug. And we manage to kill off the orange cells at the top, we kill off the red cells, we kill off the purple cells, we kill off the blue cells. But just by chance, the yellow cells you see happen to acquire a mutation before we began treatment that render them resistant to this drug. So they're able to pass through the selective bottleneck that we've introduced. And nevertheless, we kill off 99% of the cancer cells in the patient. Tumor shrinks, patient gets a lot better, but he returns 6 months later with relapse because that population of cells that passed through this bottleneck was able to reseed the tumor. And so the patient comes back in the clinic, is very sick, and we need to retreat. Now, it's worth noting at this point that the mutational process actually accelerates once cancer begins, because cancer wants, insofar as it has a will, to explore mutation space as fast as it can. So it'll knock out a lot of the mechanisms that maintain information fidelity in the genome. And so the cool thing is that we can actually recover this process because there's a record of it written in the cancer genome. And I'll show you how we computationally reconstruct that. So ultimately what we want is to build a tree. What I've shown you so far already looks a lot like a tree. What we want to build is the tree that you see at right. So specifically, there's 3 things we're interested in. The first is what genetically distinct populations exist in the patient. And the cool thing is that we can actually recover ones that existed historically, even though they've been driven to extinction in the patient today. The second thing is what are the relationships between these populations, which evolved from which. That, of course, is given by the tree here. And we want to know how much of each of these populations is represented in the cancer. So this becomes really important in the clinic. Not only does this inform our basic understanding of cancer, but we can change how we treat patients based on this. So what you're seeing in the cancer is that, let's imagine, in the tree at the right, we had this initial population 1 that started the tumor growth from this descended population 2. But then from that point, given the mutations that the cancer was acquiring, it started to explore 2 separate paths through mutation space concurrently, represented by the 2 branches. So the top branch, which has populations 3, 4, 5 in it, is interesting because the colors are showing what proportion of each population was present at 2 different time points, first at diagnosis and later at relapse. So because all those nodes in the top branch are blue, it's saying that they were extremely well represented at diagnosis. They constituted essentially the whole tumor. But by relapse, there's no red. So they were driven to extinction. The bottom branch, however, is telling you that because you see just a little bit of blue in node 6, that this population was just starting to grow at the time of diagnosis. There was maybe 10% of the cancer cells in the patient. But the interesting thing is that when we treated and we managed to kill off the entire top branch, the bottom branch was left intact because it was resistant. And so it is this branch that gave rise to relapse, which is why you can see nodes 6, 7, 8, 9 all being extremely red. So if we could have predicted this tree at the outset, at diagnosis, we could have treated to try and drive to extinction both of those different branches. And we potentially could have prevented relapse. So how do we do this? Well, we start with tissue samples taken from the patient. So we're going to get a mixture of all the different types of cancer cells in that one patient. Ideally, we get multiple different samples instead of just one sample, because that confers more information. So these can be different samples from different spatial locations. You can imagine we got biopsies from different locations in the tumor. Or they could be samples taken at different points in time. So that would be like the diagnosis relapse example I was just telling you about. We feed this into our genome sequencer, which gives us a readout of all those 3.2 billion letters in the genome for the cancer cells. And the data is challenging. It's noisy, but it's also this convolved signal from all the different types of cancer cells you have, as well as some contaminating not cancer cells. And so if we're going to do anything with it, we have to try and deconvolve this. The final step is to do mutation calling. So this is just telling us exactly which of those letters in the genome have changed between the normal and the cancer cells. This is all done computationally, but it is this signal that we use to build our trees. Specifically, we're interested in the frequency of mutations. So what you're seeing here is every row in this matrix corresponds to a different mutation in the patient, and then the 2 columns correspond to our 2 samples. The right sample is diagnosis, the left sample is relapse. The intensity of green in each cell in this matrix is telling you exactly how prevalent a given mutation was in that sample. And so that's starting to look a little bit like our tree, because you can see that for, the top 10 or so rows here, they have mutations that are extremely prevalent at both diagnosis and relapse. That would be like nodes 1 and 2 in our tree. Then below that, we have another 10 or so mutations that are prevalent at relapse but not diagnosis. That would fall somewhere on our bottom branch. And then at the very bottom, we have some mutations that were prevalent at diagnosis but not relapse. So that would be more like our top branch. So the way that we go from our mutation frequencies to a tree is where our computational stuff enters. So these frequencies confer quite a bit of information. Let's say I gave you 2 mutations, mutation A, mutation B. I have 5 distinct tissue samples, samples 1 through 5, that give me frequencies of those mutations. Now, you always inherit the mutations that your predecessors had. So that means that because we see mutation A is prevalent at a high frequency in all 5 samples, mutation B is absent in 3, that means that A has to be an ancestor of B. So whatever tree we build has to have A on a path somewhere upstream of B. Similarly, we can infer these different branch relations. So we can say that, again, because you inherit all of your predecessors' mutations, because A is higher than B in some samples, but B is higher than A in others, neither A can be a parent of B, nor can B be a parent of A. So we have to put them on different branches. Now, if we enumerate this, we see that there are actually 4 different possible relations any pair can have. We already talked about cases 2 and 4. So, of course, we also have case 3, where B would be ancestral to A, and we have a case where mutations would exist in the same population of cells because their frequencies are about the same across all the tissue samples we observe. So what we can do is figure out a distribution over relations for every pair of mutations. So this matrix is showing you exactly that. Every row and column correspond to a mutation. It's a square matrix, so it's just telling you what the most likely relation is based on color for any given pair of mutations. So, for example, this big blue block that you see at the upper left would be mutations that should all be clustered together in one population of cells. Whereas the green rectangle to the right of that corresponds to mutations that would be ancestral to, that would be a descendant of that initial cluster. So again, any tree we build should satisfy the relations that we see here. So we can just do MCMC to recover a tree that satisfies those relations. So we've already figured out what mutations should be clustered together into single cell populations. So out of 50-some mutations, we end up with 11 distinct cell populations. So there's one or more mutations that differentiate every one of these populations from the others. We can start with this fully branched tree, and then we just make metropolis proposals where we'll detach a branch and move it under a new parent. Then our likelihood is based on how well the relationships implied by this tree match the relationships implied by the data. Do that 10,000 times, and you end up with a tree that looks quite reasonable. So the last step, as you'll recall, was trying to figure out what the frequencies of these populations were in the cancer as a whole. We want to know how much each population is represented in the cancer. So, of course, the data we have already strongly hints at what those frequencies should be. The catch is that they have to be tree constrained, because the sum of frequencies in children of a node must always be less than or equal to the frequency of the parent across all the different tissue samples we observe. So it's easy enough to define an objective that we can just do gradient descent on so that we get values that match the data as well as possible while also obeying the tree constraints. And so the figure all the way at the right is just showing you the same information that exists in the tree at center. It's showing you that you have this one initial clonal population from which everything spread, and that you had this diagnosis-specific branch, and then you had this relapse-specific branch. So that's cool, but what's a lot cooler is the work that we've been doing with Stephanie Dobson. So she brought this wonderful data to us. She's a biologist, where she actually has 95 distinct tissue samples from a patient in some cases. I don't have time to tell you about this now, but if you're interested, seek me out later and I'll tell you all about it. But this gives us a great deal of information, and we can recover a lot of resolution with respect to how the cancer evolved. So I hope that I have shown you three things. The first is that you can look at cancer as an optimization problem. The second is that we can do this genetic archaeology to reconstruct the path that cancer took through mutation space. And the third is that from this, we can get not only insights into cancer biology, but we can also better treat patients. So I just want to acknowledge the Morris Lab in particular. The folks at the top are the ones that are working on cancer. The work I showed you today was a collaboration just between me and Quaid, but everything's been driven by what Dr. Dobson brought to us in conjunction with her supervisor, John Dick, at the Princess Margaret Cancer Center. So it's been a wonderful collaboration for us. Thank you. I'll take any questions.